Well, hello and welcome to another episode 17 of the Wondrous Stuff Vodcast, broadcast live on Google Hangouts on air every weekend, Sunday as usual. This week. This is the place where you'll find news, information and commentary on science, engineering and technology from the past week and beyond, and anything else that interests us from inside our tiny little human brains. As always, my name is John Gardner, and to help us out on the journey to knowledge, we've got my colleagues Richard Smith and Ross Davidson. Say hello, chaps.
Hello, chaps.
And this week, for the very first time, we have a genuine scientist live, and his name is Jack Williams. Hello, Jack.
Hi there.
So, perhaps you can start off by telling us how...
Turn your camera on, John.
Yeah, I was going to say, you're just a giant brain at the moment.
My camera's on.
Can't see you.
Yeah, no, camera's definitely on. I can see me.
We'll use our imaginations.
Okay.
Anyway, so perhaps you can start off by telling us how you know Ross really, and how and also a little bit about what the work you do during the day.
Right, yeah, okay, day and night, 24/7 here, I'm afraid. And I've known Ross since I moved up here to the northeast and met him in a pub, shock horror. So, and over the years we've had many interesting conversations, some about science, a lot about football, and a lot of, well, just gibberish really. Richard I met through Ross, and even though I've not met John as such, I'm aware of his presence. So, what I get up to basically, I'm a biomedical technologist who specialises in blood. I've covered a lot of fields over the years. I started off making reagents for blood testing out of human blood. Moved up and worked in the field of red cell immunohematology, which is mainly dealing with autoimmune disease and antenatal problems. And after leaving Sheffield and moving up here, they, for some reason, wanted me back again. And now I help manage the blood processing plant in Newcastle, where basically we take all the blood that comes in from all the donations in the northeast and Cumbria and make things out of it. I had one particular subject I thought I might have a bit of a look at today, which might be of interest, in that most biological molecules we seem to be able to produce artificially in some form or another, but nobody's ever been able to hit on making a decent blood substitute. I'll hit on the reasons why in a bit, but it's actually incredibly in demand at the moment due to all things like HIV and all the rest of it that's come along over the years, and also because we have a drop-in donor database, so it gets more and more difficult to get blood out of people. Okay, that enough for a starter?
Yeah, well, yeah, go ahead. I mean, just so you synthesizing blood, and that was your main core, wasn't it? And
sorry,
why
John.
It's not easy to do that.
Yeah, I was going to talk a little bit about that. I'm not going to get too technical because I'm aware that I'm the token biologist here. It's something that seemed to be beyond us for a long, long time, which is the production of blood substitutes. And I mean, the basic reason why we can't produce anything is hemoglobin is just a horribly complex molecule. Hang on a minute. Does that come up?
Yeah.
There we go. That's hemoglobin, which I'm sure you're aware is the actual chemical that's in blood. Interesting little chemical formula there. And what hemoglobin does is it picks up oxygen where it's in a high pressure, high partial pressure of oxygen. It links in within the molecule onto those little, what we call the little 00C molecules that are on the end there, and then gets carried. What's in hemoglobin is actually 4 molecules of heme, and the iron molecule in the middle changes from ferric to ferrous state, Fe2 to Fe3. Fe2 when it's holding blood, and then off to Fe3 as it releases it, and it's all based on how much oxygen is there. So it picks up the blood in the lungs, the oxygen in the lungs where you've got absolutely stacks of the stuff. Even if you've had some sort of injury or lung disease or something, even at 85% capacity of the lungs, it still works ridiculously efficiently, and then just drops it off when it gets to somewhere with a lower partial pressure in the tissues. basic point about why it's been so difficult to synthesize it, well, it's quite obvious. Look at the molecule. Getting stability on that artificially is a nightmare. So I just thought I'd look at some of the other alternatives. Hang on, let me come off that screen share. Because I've ever done that. There we go. Right. So there's been attempts to try and produce a blood substitute since the 60s, and it really accelerated during the Vietnam War. The Americans had basically tons of cash and lots of dead people. So what they wanted to do was get something they could get out on a battlefield, which had very little accessibility to it, and get out there and actually treat people on the battlefield. There's also other big pressures over it over the years. I've got figures from the States here, unfortunately. Even though I work for them, I couldn't find any UK figures. Donations are only increasing about 2 to 3% annually, but the actual requirement is going up at twice that rate. We've obviously had lots of problems with HIV, particularly in Africa. Nigeria is a standout country. I mean, it's something like 40% HIV positive in the country now, and because they're a fairly skint country, unless you happen to own one of the oil wells, a lot of the blood isn't actually tested before it's given to people. So if you're travelling around in Africa, Kenya, for example, don't have a transfusion.
I was going to say it's the second largest cause of new HIV infection is transfusion as well, which is really quite scary.
It is. Yeah, it's absolutely terrifying.
So they just don't have a good scanning program in.
It's bloody expensive to test blood. And the other thing is with HIV, hepatitis B, hepatitis C, hepatitis, I think we're up to J at the moment, but there's another one getting added every couple of years or so. The big problem we have with it is we have what's called a window period, where basically you've already picked up the disease in your body, your immune system started to produce an attack on it. In the case of HIV, that's very difficult with it being a retrovirus, but you can't actually detect it. So most of the tests out there that I deal with, which is blood, don't pick something up until you've actually been infected for a good couple of months. That's why we screen very heavily on lifestyle in this country. But having said that, if you're desperate, you'll take out. You take HIV-infected blood if it's a choice between that and dying anyway. I mean, there's other reasons for it. Blood's quite a swineless store. In this country, we only keep it for 35 days and it goes in the bin because the cells start to break down, and we also get an increase in nitrous gases in the blood, which can be quite harmful. If it's left for much longer than that, you can actually get the bends from a blood transfusion, which I can imagine would be not good if you're fairly ill anyway.
So a synthetic would have a much better shelf life, presumably, then?
Yeah, I mean, I'll come on to that. I was just clearing up some of the reasons why we're after it. I mean, there's also the fact, as you're probably all aware, blood comes in 4 main groups, O, A, B, and AB, which cuts down the problems there. They're the main ones we worry about. They're the big killers. But also you've got the RH system, which some people refer to as rhesus. But it can't be rhesus. Rhesus is a monkey. It's actually the RH system. Which you'll have seen D positive and negative, but there's actually 73 different blood group systems out there, and the chances of getting an exact match are something like 1,400,000 to 1. So there's a risk inherent to all times, and some of these are, first time you have a transfusion, not a problem. Second time, you're dead. I know this from experience, having missed one many, many years ago. So anyway, stop rattling on that area. What substitutes are there possibly available? The main one that people have been looking at recently, I say recently, they've been looking at it since the 70s. It's been produced by Oxygent and Oxycyte, both American companies. Massive target for this in the States. Is perfluorocarbons. Has anyone heard of those? No, right.
It's a new one.
Okay, it's just starting to be used in diving, high saturation diving and things like that. It doesn't actually mix with blood at all, so you have to make an emulsion of it. But it's carrying quite well. The problem they've got at the moment is so phenomenally expensive to produce. I've lost my bloody diet. I actually had a formula of that. That's the main one we're looking at, but at the moment, because it doesn't dissolve in blood, it's a bit of a nightmare. The other problem with the other side is artificial hemoglobin. Now, the biggest problem you've got with hemoglobin is it's poisonous, which I know sounds counterintuitive, but hemoglobin breaks down the bilirubin the minute it hits the liver. You go bright yellow and you die of jaundice. Hemoglobin has to be kept in cells, and cells, nobody knows how to artificially manufacture cells yet.
So it's basically because the hemoglobin is in the center of the cell that it's fine. If you take it out, that's when it becomes poisonous.
Yep. Basically, you end up with the plasma, the sort of liquidy bit of your blood, to be technical, goes a nasty brown colour, and you get a thing called bilirubin. Basically, your liver packs in, which, obviously we try and avoid that wherever possible, apart from on the long-term basis. BioPure in the States seems to be the biggest company doing this at the moment, but there's no successes yet. I'm a bit iffy about some of the stats here because I wouldn't have published this. They're too woolly. But in the use of the HemoPure, something like 36 out of 350 patients died, as opposed to the control. These are too small numbers for me to be that relevant, to be fair. 45 out of 350 died. Now,
this on military personnel in the US, was it?
This is terrifying. This could only happen, well, in a dictatorship or the States. They didn't actually tell people they were doing it.
Yeah, I remember.
On patients. This was on general patients. And they didn't tell them.
Wow.
Sorry?
It was on general patients, wow.
Yeah, I know.
I'm surprised they didn't get away with
that. Well, they didn't. 45 of them died.
Well,
I'm
yeah.
just surprised.
Like you say, only in the States, yeah.
Yeah, well, the most litigious company in the world as well, but apparently the government gave the Food and Drug Administration permission to do the trial without telling anyone. I like to think that would never ever happen over here, but who's to say nowadays,
What was the death toll? 40-something out of 300 and something, was it?
Yeah, I mean, it's the one worked out at about 10%, the other one worked at 10-11%. I mean, I'm no expert on statistics, but I wouldn't say there was different.
You'd need a larger group to see how significant that is, wouldn't you?
Yeah, well, the first thing you need to do is tell people you're doing it.
No one's going to volunteer for that, though, is it? It's hard to take transfusions when you know when you were saying before about it's interesting about transfusions. Though, have you this story about this wrestler who died last night? Did you see that? The Mexican
I didn't see that.
The Mexican one, I saw that. Yeah.
Yeah, and seemingly he refused a transfusion even though his life was. Was it him, or was it something else I was reading about? But anyway, you'd think that in a life-saving situation, you know, when you were saying before about HIV-infected blood, you would just go for the transfusion. But it's remarkable how many people actually, not just Jehovah's Witnesses and religious people who can't take it, but, people refuse for all sorts of strange, logical fallacies, basically, like argument from nature and things like that.
Yeah. I like the term logical fallacy. I'll remember that one. No, I mean... It's similar to people refusing drug treatments because they'd rather have homeopathy or something like that. I sort of have trouble understanding these people, and I think unless you're one of them, you're never going to. I mean, there's a few other alternatives out there that are interesting, but this is sort of getting into the realms of weird and wonderful. Hyperbranched polymer protected porphyrins, which it sounds brilliant, but it's actually creating artificial blood from plastic.
Ah, okay.
Yeah.
So when you start about creating an artificial blood,
yeah,
presumably you have to. I mean, the sort of, I guess, blood's made up of things like red blood cells, white blood cells, platelets, plasma. That's all part of blood, isn't it? Has it all have to be created artificially, and have they managed to do any of it artificially? Any of the parts, the constituent parts of...
The only thing I'm aware of, and I've been working in this field for 22 years, the only thing that we've done is bulk people out with saline in place of using plasma.
Water it down a bit.
Yeah, basically, salt. You give them some brine to keep them going till you get them back. If you watch Casualty or other medical programs are available. But if you watch something like that and they're always hanging the bag on the bit of string on the person next to it, it's normally just saline. And what you do is you do fluid replacement, just to keep people moving, because obviously if you've lost too much blood, your lungs start to shut down and things like that. And all they're doing is bulking you out. If you're not actually doing anything, you can survive.
Is that to get the pressure back in then, is it?
Yeah, pretty much. Yeah, it's keeping the pressure up. I mean, the platelets are horribly complex, and I've deliberately avoided going into those because it's an area that confused me for many years. I mean, for starters, platelets are dead. And they're set off, I think it's just a couple of interleukins, and then you get a chain that makes them cause the clotting effect with the fibrin, which is in the liquidy part of the blood, to be technical again. White cells, once again, there's so many different types and things, and they respond in so many different ways that to produce them artificially. I mean, the one thing that is coming out, and I was going to get on in a bit, but I'll hit it now because it sort of comes in at the right time. It's been looking at the possibility of using stem cells to grow blood in a laboratory. Obviously, are you much aware of stem cells and what they're capable of and what they are?
They seem to come up a lot in a lot of really revolutionary sort of like treatments and stuff. They're definitely something that has a lot of potential.
I'll do a quick 1 minute explanation, and that'll probably be most of my knowledge on stem cells gone nowadays, because I've only got so much room in my brain, some stuff has to go. Stem cells basically are the precursor for every cell. It's what we call a plenipotentiary stem cell, and from that, the theories go, and quite a lot of the experimentation backs this up, you can grow anything. You can then turn that cell to everything from a sclerotic cell, which is a nerve, all the way through to a blood cell, a brain cell, and everything. What people are working on is how you cause that to happen. And it's a massive area of research at the moment. And there's people getting on, really down the way and managing to turn them into other things. There's things, replacement for bone marrow. The really big area of research is growing nerve cells, because they're fragile. Once they're broken, they don't repair. So, if you could come up with some way of regrowing a nerve cell, it's the sheath of the nerve that's a bit that causes the problem. Basically, all those poor people who've broken a back or got a smack in the neck or whatever and have lost the use of part or a lot of their body could technically be up and walking again. Yeah. So, massive research there. There's other potential techniques which I won't go into too much because they are just odd. Things like placental umbilical cord blood. I've actually been involved in this in the past. It's collecting what we call the cord blood from the placenta. It's birth of a healthy baby and using it as a blood substitute, because trying to get into this without getting too far away. Basically, nippers have a different kind of hemoglobin that is more thirsty for oxygen than normal adult hemoglobin. HbA is adult, HbF is fetal, and fetal hemoglobin will carry a lot more oxygen, and it will steal it at a lot lower partial pressure. So that's another potential area.
That's the one that they were doing on the US military. I remember now. Because you can produce, like you're saying there, with the umbilical cord, you could produce so many litres of usable blood, couldn't you? They're having quite a success for that. But obviously then it's the limited number of umbilical cords that you can get access to.
Well, the ethics are incredibly tricky with this sort of thing.
So, quick question. In the body, where's blood actually produced in the body? Because I may be wrong here.
Isn't it?
When you give blood, you give a pint or whatever, and then your body replenishes it.
Yeah, so fairly rapidly.
Where's it produced in the body?
It's in the bone marrow.
Yeah, it is in
the bone marrow.
Yeah. When you're very small, all of your bones produce blood cells. White cells, red cells, or the plasts that become platelets.
How fast does the body produce blood? Like, if you were going to give blood, how much blood could you give without it affecting?
Well, I mean, we take around a pint. I hate using imperial measures. I actually worked it out. It's 610 ml. It's ideal. We take about that much because that's the fluid in it's replaced almost immediately. So obviously your body just doesn't excrete so much fluid. The cells have a 35-day life in the human body as well as in a pack.
So every 35 days your body replenishes all your blood anyway?
Yeah, it has to. It dies. That's largely what you're whittling out quite a lot of the time is products of blood breakdown. So yeah, 35 days, but you've got to remember there's other things there. The platelets, it's about 7 days. I'm a platelet donor. I go in for a thing called apheresis, where they take, I think it's about 200 ml a time off me, spin it down in a machine when it's there, take the platelets and the plasma off, resuspend my own blood cells and give it me back through the same needle. And that's it takes about 2 hours, and I go do 10 cycles of that, and that's just a marvellous sort of platelet. And I'm not a very rare blood group, but I'm the one that's most required for platelets at the moment. So,
what is your blood group? What is the rare one, or what is the one that's needed for platelets?
A negative, we're absolutely in demand with them. A negative platelets can usually be given to pretty much anyone with no ill effects whatsoever. So they're being used heavily on what we call trauma packs now. There's a lot in the press recently that North East Air Ambulance is starting to carry a trauma pack with it at all times, so they can do, side of the road. It's quite often side of the road, or I don't know, bottom of a cliff or whatever like that. Transfusions on the spot, but obviously they can't carry a full load.
This is the ones where the blood bikes are coming in and transferring the packs off to the air ambulance.
Yeah, that's right, yeah. And the hospitals are starting to keep trauma packs, so if somebody comes in, back of an ambulance in a state, they don't have to bother blood grouping or anything like that. They can basically get the person stable while they wait for the consultant to wander down and get involved at that point and get bloods done, actually start to ascertain people's blood groups and things like that. So it carries what we call universals, which is A neg platelets and O negative. I nearly said O rhesus negative, then got myself in trouble with the people at work. O negative blood cells.
So not wanting to go off on too much of a tangent, but on that, in terms of the blood groups and the sort of compatibility, what's the reason that some blood groups are compatible with everyone and some aren't? Is it just to do with the structure of the cells? Or?
No, it's to do with naturally occurring antibodies. I'm Group A, so I naturally have anti-B. So if somebody puts some Group B blood into me, I'll basically break it down, and it will set off such an immune response that it could kill me.
So your body just rejects it.
Sorry?
Your body just rejects it, basically.
Yeah, pretty much. That's the easiest way of putting it. But what happens is it breaks it down so quickly that the breakdown particles hit your liver, and that's what kills you. As I was saying earlier on, it breaks the cells up. The hemoglobin comes free, and hemoglobin's a poison. That's Group A. I mean, somebody who's Group O has anti-A and anti-B, so obviously they can only have Group O blood. If you're unlucky enough to be Group AB, you don't have any of the antibodies.
That's me.
Oh, yeah? All right, you're dead easy then. You're no problem at order transfusion.
Yeah, I'm AB as well.
Yeah, Christ, the odds on that actually are quite low of having two Group AB people here.
I looked it up because Susan is as well, which obviously means that the kids clearly are going to be, aren't they?
Not necessarily.
Alright, so if we're both AB, potentially the kids could be something else.
The kids could be A or B. They can't be Group O, but they could very easily be Group A or Group B.
Alright.
So AB is the universal recipient, is that right?
That's what it's referred to, yeah. There are still risks, but, that's what... The main killer in terms of transfusion is the ABO blood group system. The rest of them, you generally, you can get away with it once.
So I'm O positive, I think.
You're O positive, right? Yeah,
so
yeah.
I'm a universal donor, but I have to have O.
Yeah, O positive. Yeah, in terms of the ABO system, you're quite handy to have around. Fairly common in the northeast, about 45% of the population. John and Richard, you're down in the 3% area. The demographics change depending on how far north you go in the country.
Why is that then? I've understand across countries in different continents and stuff, but just in the UK, there's a significant difference.
Yeah, it's not massive, but it's spottable. We're talking in the terms of like 3-4% between the north and the south, and it's basically the people in the south have got a lot more Roman in them, and people in the north have got a lot more Viking and Pict. To put it in very simple terms.
Yeah, and if you do a genealogy, you'll find surprisingly that people don't move. I remember me on my maternal side, they've got a very unusual surname, so it was very easy to do the genealogy. And there was only one person who left the Northeast, basically. The rest, going all the way back, just stayed where they were. So populations, like I say, from those times have just stayed static.
Yeah, I mean, we've
on that Roman thing. Interestingly, Centre for Life were doing a thing where they contacted Robsons, people with the Robson surname, men with the Robson surname, because they were trying to prove or disprove the hypothesis that there was a common ancestor from Roman times amongst a high volume of them, and they were right. Something like 85% of the Robsons that they brought in all had the same ancestor from whatever 1800 years ago or something like that.
There's a lot of DNA research going on at the moment to do with looking, where people were from originally and things like this. And I must admit, I've been tempted to go and have it done myself. I believe there's a fee, but I think it'd be worth it. So because you can trace certain areas of the genome, and you can trace them to area, you can actually trace almost how your family moved across Europe, or didn't in some cases, the Robsons, obviously. But you can trace how people have moved by their genetics. And it's not really my field, but it's something that's quite interesting. But they've managed, I assume, from taking samples from people from graves and things like that back then. But they can actually trace your DNA and where the movements of your ancestors has been over the years. It's interesting stuff.
They should make UKIP voters take it.
No, actually, I'm trying not to be political here. I'm not going to comment, but those of you who know me know what it's going to be.
Did you hear that news? I think it was maybe about 2 years ago. There was a person on Oprah or Jerry Springer or one of those kind of shows over in the States. It was like a massive bigot, especially against blacks, and they forced them to have a DNA test. That is like even though he was to look at Caucasian, his like a great grandparent, not as far back as you would think, like a great grandparent was a black African.
I bet he was happy
challenged his view.
Yeah, it's always nice to challenge people with very tiny minds like that. I really enjoy doing it. There was a thing many years ago when I was living in Sheffield. My darling wife showed me in the shop, somebody was handing leaflets out where they would give you dietary advice based on your blood group. I don't know if any of you heard of that.
No, but
is there any science in that?
There's the theory behind it. I use the term theory in the loosest possible sense. Is that the blood groups did evolve at different times. Off the top of my head, I can't remember, but I believe O was first. Ross, you're a bit of a Neanderthal. And AB was complex and came down longer along the way. So some smarty pants had come up with the idea that Group O's would have been hunter-gatherers. Basically, Ross should eat a lot of meat to get the correct diet for his physical type. Whereas
that's good because I do.
Yeah, okay. And obviously, John and Richard, you will have been more towards, sort of agricultural side of things, so you're probably better off sticking with a lot of fruit and veg and stuff like that.
There you go.
It's funny you should say that, because it's true. Yeah, exactly. There's 3 vegetarians on the panel, and I'm the only meat eater.
Oh, right, I didn't realise that. But, yeah. To be fair, the thing that bugged me about it was they were actually doing blood group testing in the church halls and things. They were doing this. They'd obviously bought some reagents online from the States, and they were basically, prodding needles into people's fingers and then giving them advice with a very, very tenuous link, shall we say, to reality. But I've heard quite a lot of these things over the years, and they are quite fascinating. And if ever you get the time, it's always great to go to one, particularly if you've got either you've got the time to read up on it, or this one was quite random. And it's my field, so it was quite funny to go and shoot the guy down.
I've seen lots of similar things to do with various dubious people coming in and saying that we can prove that you are allergic to X, Y, and Z by putting a little bit into these electronic supposedly electronic machines that they buy, and you hold one anode and you put something in the other thing, and it's like,
I've seen that.
yeah, that's just
I think
rubbish.
I think I've heard about it. There's so much, absolute nonsense science out there. I mean, I was just flicking through on the wondrous stuff earlier, and there's that young American lassie going on about why dinosaurs couldn't fly because their eyes are too big. I actually find myself I have to turn off sometimes now because I'm quite a positive person about the human race in general, but then you see something like that and you're starting to think, well, how long is it till the cockroaches take over?
So, one question I've been meaning to ask on the subject of blood groups: do other mammals have different blood groups as well?
Yes. I was involved just on a periphery a while ago with setting up a dog blood bank. Dogs actually have 2 blood groups, and it's a very expensive thing to do for dogs for transfusions. But as somebody who shelled out £2,500 on a dog getting a dog cured of cancer over the last couple of years, I sort of I can go along with that. But yeah, dogs have 2 blood groups. Just positive and negative to keep it simple, but the problem they had with setting up the dog blood bank was a lot of people were willing to, come along and get the blood taken for the dog, but you turn up with a chihuahua, it's largely pointless. So they were actually asking people to bring, I want your Rottweiler through the door or something,
What about things like the great apes and that sort of thing? Do they have blood similar to us? Is there a possibility of like transfusions from, I don't know, pigs, things that are compatible with us? Could that work?
In a word, no. Basically, it's nice and simple.
Yeah.
It's been tried. I mean, the first, let me just get one of my references up because I've read that much today, I've forgotten exactly where I'm at. Things that we used to mimic blood originally, where are we? Where animal blood. It was done it's a classic like woodcut of it back from the 17th century, the first time a transfusion was done, and in that case they used a lamb, I think it was, and the first time the guy was sick, it was absolutely fine, no harm done at all. They had no idea of how immune systems worked at the time, so they tried it again on this guy had volunteered, and the second time he was incredibly ill, and I can't remember off the top of my head whether it killed him or not, but I have a suspicion that I think it did kill him. Because obviously the first time his body dealt with it because it had never seen it before. Second time round, massive immune response, power dead. One of the things you can see from that, one of my favourite medical terms for years is disseminated intravascular coagulation, which basically means your entire body clots. And I've never seen it happen, but well, it's painful for a very short period, I believe.
So is there any species of animal where the blood is near to a human blood?
All the mammalian blood is similar, but the blood groups are actually there. What gives blood groups on the membrane of the cell is they're basically the channels that oxygen goes in and out of the cell for. And they've evolved into so many different, a group A person has basically different oxygen tunnels to a group B person. I won't go into all the chemistry, A, because I can't remember it that well, and B, I seem to remember it being quite phenomenally boring when I was studying it. But it's all basically down to the way the channels within the blood cells are. But the basic hemoglobin carrier molecule, the red blood cell, isn't that different until you get down to some of the other forms of life. I'm trying to think, it's a certain crabs in general have blue blood, which is cyan methemoglobin. And I believe it's a cyanide molecule instead of an iron molecule in the centre. Well, it seems to keep crabs going pretty well. Evolutionary, they seem to be quite aptly trotting along sideways. So yeah, there is other forms. Well, in humans we have several different kinds of DNA, as I sort of got on earlier. There's one you may have heard of. Have you heard of sickle cell disease?
Yeah, sickle cell anemia, isn't it?
Yeah, that's right. I mean, sickle cell is quite an interesting one. The basic what we call sickle cell traits, sorry, but go back a bit. You basically have the 2 genes for, two presentations of hemoglobin. In most people, such as ourselves, you've got fetal hemoglobin twice. Sorry, adult hemoglobin twice. You have fetal when you're born, it switches over to adult hemoglobin, very sharpish. In sickle cell disease, you haven't got HBS, you've got this thing called HBF. Now, if you've got a double dose of HBF, you have sickle cell disease, and that's transfusion life. You know, it's not suitable for life. However, if you have sickle cell trait where you only have one dose of HBS and one of HbA, you can generally survive perfectly well with that, and it won't be noticed unless your body's put under very high pressure, basically low partial pressures of oxygen, which happens when you go on an aircraft or you go up to a very high height. And what happens then is the sickle cells, hence the name, actually change shape and they block the capillaries. The interesting thing about that is, because it's a mutation on the cell wall, is that in Africa it actually protects you from malaria, because the plasmodiums that cause malaria can't travel through HbF. So if you've got the mixture of the disease, it's actually it's evolutionary prominent if you happen to live in an area where malaria is quite rife, which is why it's still here. Because otherwise it probably would have bred itself out. Even recessive conditions will breed themselves out over time.
And is the sickle cell disease, is it more prevalent in Africa?
Oh yeah, very much so. Yeah, and the current theory is that it's more prevalent there because it actually is an advantage to survival.
So that is a pure genetic trait that's part of evolution?
I imagine that it's been around in most populations, but in the rest of the world where it was, it's a recessive condition, it's not giving you any advantage whatsoever, and it's spread itself out. You know, whereas in that area, because it does give you a slight advantage, that's why people believe it's still there.
Wow.
Yeah,
That's
not directly my field, to be honest.
No, it's all interesting stuff.
So has anybody here had a blood transfusion at any point?
My son has, and my wife.
Yeah.
Both on the same day, funnily enough.
I've got people in my family who've had blood transfusions, yeah.
Yeah.
Actually,
Just
my wife's parents, both my wife's parents have had blood transfusions.
Just out of interest, how many of you donors?
Donors? I am a
donor.
donor. Yeah. Although I have to say I haven't been for about a year and a bit, but I am a donor.
I'm booked in for one shortly. You'll be happy to know.
Depends on how busy I am that day at work.
My fifth one, so I get my next card. You get the 5 to something card after that, don't you?
Yeah, I'm not going to be over.
I'm on my silver card.
Yeah, I'm not going to be pious. I've just gone on to donating platelets, and which I can do every 4 weeks. But that was my 3rd donation for 20 odd years, and so I can't really say anything about it.
So does
I used to be a donor, but the last time I went, which was probably about 10 years ago, I think probably because I'm the universal donor or whatever. I was actually told that they had too much of that.
It can be the case.
They didn't need it. So I mean, that was obviously 10 years ago. I don't know what it's like now, but I would imagine that, again, in percentage-wise, is that the O is the most prevalent anyway.
It is, yeah.
You naturally would have more of that anyway.
The thing is, we've got more, it is the most prevalent type. It's very close between O's and A's. It is the most prevalent type, but that means there's one hell of a lot of people who want it.
I suppose so, yeah.
So when you go to give blood and that all blood is all taken away, is it stored regionally or is it stored centrally within the
Right,
country?
I mean, this is exactly what I do at the moment. I deal with the area where we manufacture stuff out of the blood. And you give your unit of whole blood, and say you go to the annex session, for example, that comes into the area where I work by about 9 o'clock that night, and we're straight working on that. And if it's not done that night, it'll be started on the next day. And the first thing we do with the blood is we pop it into a fairly fast centrifuge, cryofuge, keeps it at 4 degrees, or 3 degrees, depending on what we're doing. I won't go into the details. And basically we spin that down, so you've got the red cells on the bottom and the plasma on the top. And depending on what bag, you might also have what we call a buffy coat in the middle, which is concentrated white cells and platelets. What we do with that is we then chuck it on what is basically a press. Bear in mind we're doing, up to 600 of these a day. We put it on a press. We push the plasma off onto another bag, which if you are female, we generally don't do anything with it. If you're male, we will generally freeze that down into a separate product called fresh frozen plasma, or we may make something called cryoprecipitate. But basically, they're for clotting factors we're after there.
Hold on. Sorry to interrupt. There's
That's
male
all right.
and female.
Right, yeah,
Okay. Male and female blood.
No, it's not a difference with the blood. The reason why we don't tend to use female plasma is during pregnancy, and bearing in mind this can be a pregnancy that you may not even know you've been pregnant, and then it's miscarried. You get neonatal antibodies built up, which can be harmful to some patients, particularly if you've got a depleted immune system. Somebody, a very young child, or somebody who's undergoing some sort of immunotherapy or something like that. So we don't tend to use female plasma. And there was some talk about people were saying, well, why don't you take it from females who haven't been pregnant? That's because you may not even realise you've been pregnant.
Yeah, it's just safe. It's just a stick.
It's a safety thing. Obviously, we're very safety aware. We're making medicines, so we have to be very, very careful. So so the plasma gets taken off. The red cells then gets resuspended in a solution of saline with other things in it, basically to give the blood something to live off. Red cells something to live off. And if you've donated regularly, Richard's probably doing this being a male, probably doesn't realise it, we will also, out of the middle bit, what's called the Buffy coat, take that, and we will then put that through another process to extract Richard's platelets, so that we can make a unit of platelets from that. Platelets only have a 7-day life, and we are always desperate for platelets.
So when you were a platelet donor like yourself, how is it? You said it was a two-hour process. Does it hurt particularly, or is it just the same process as you?
I mean, you feel a little bit of a prick when they put the needle in. It's not painful or anything. It's an odd sensation because there's quite a lot of pressure on these things that they use, and you can actually feel the blood being taken out of you. Nobody's squeamish here, are they? No. But you can feel the blood being taken out of you. It goes in the machine, and you can actually feel it coming back a degree or two cooler, which is quite a weird feeling, to be honest. But
is there a national register like there is for? Can you register to become a platelet donor?
Oh yeah. That's why I did it. I mean, I went in, gave a unit of whole blood. The lovely staff in the donation suite, because you have to go to Newcastle to do it. They're quite big machines and they don't travel very well. Saw my blood group and went, would you like to become a platelet donor? I'm like, yeah, I was hoping I would be. So then you get in and you'll be called in every 4 weeks or so, because it's the most demand product out there. So yeah, that's a separate panel, and there's all sorts of other panels. There's panels for things like the blood bone marrow registry, which I'm on. There's the blood cancer one that I've seen on Facebook a lot recently, and that's for donating your bone marrow. That's for people who have usually had whole body irradiation because they've got some sort of leukemia or something like that.
That's supposed to be quite a painful process, isn't it?
I believe so. But you're going to save somebody's life at the end. Just for the case of a bit of a painful process, I'll do that.
Mm-hmm.
I mean, that's what they're trying.
We know somebody who's going through that, don't we, Ross?
We do, yeah.
It's probably the same person I'm thinking of as well.
Mm-hmm.
It's a good thing to do. The other advantage of it is you find out what your histocompatibility, basically your tissue type is. And that's always interesting to find out, because I found out I had an incredibly common tissue type, which means there's a good chance there's a donor out there for me if I need anything. No need to grow that child up in the third world anymore.
So tissue type, is that similar to blood type?
Tissue type, I mean, once again, it's histocompatible immunogenetics is something I found a dark art. I couldn't get myself into at all. I tried. But that is, the types involved for getting a match at transplantation is, to me, a horribly complicated process. I believe it's easier nowadays, but it really was just something I couldn't get my head around.
I always thought that was just pretty much blood type. You could get a transfusion, and a liver transplant or something from the same blood type, but
No, we
more
actually.
complicated than that.
Oh God, yes. We do a lot of mismatched blood type transplants now. I've been involved in a couple recently, and they need a lot of blood support, and you have to wash all the plasma off the blood because it's going into somebody of the wrong type. But there's a lot of people think that it's actually better that way, and you get less rejection if it's a different blood group.
Really?
Yeah, I've read about the mismatched transplant thing, and unfortunately I don't have it in front of me now, but it's something to do with because they've got a different blood group, it removes all the cells that could cause problems so quickly. Like I say, I'm not an expert in this area. Yeah.
So it keeps it more aggressively short term, and then long term chances are higher. Yeah, that makes sense.
This is why you see a lot of the stuff on the news and things about people growing noses on the back of mice or ears or whatever it was last time, and growing tissue in dishes and all the rest of it. I mean, there was what I can remember quite recently where they'd laid down matrix of a liver and were growing somebody a new liver within the matrix using stem cells again, which is just fascinating stuff, and I hope they get a move on with it personally.
Yeah.
So going back to the synthetics then. So you explained the challenges at the beginning then. So you're talking about hemoglobin, but really the challenge is just oxygen transport then, is it? So what our best bet is to copy biology and get something hemoglobin-like, but technically it wouldn't have to be that. It would just have to be something that...
No, I mean, the perfluorocarbons are nothing like that. I mean, they are a purely synthetic molecule. They don't mix with blood. They're just made up in laboratories, and it's a strange molecule throughout. Because I mean, you've got fluoride and carbon molecules in there, which the strongest chemical bond you can get are fluoride and carbon. It's just like absolutely as hard as hell. But I just can't seem to get my head around exactly how it's carrying the oxygen there. I tried looking at the chemistry of it earlier on, but I think maybe because it's a research area and potentially a very, very lucrative research area, there doesn't seem to be that much on how they actually work out there. I mean, if we get a decent chemist, on which I'm not, someday they might be able to explain it all to us.
So there's two approaches.
Sorry, I got two.
Potentially, you could find a sort of an artificial method of carrying oxygen that could be more efficient than blood. How efficient is blood at carrying oxygen?
Very, very efficient.
Okay.
Like I say, you can survive with very little lung capacity, and the blood will still rip the oxygen out of there, so to speak, and get it to the sites where it's required, providing you're not doing anything too hectic. And I've seen people wander into casualty units with ridiculously low red blood cell counts, and just walk in like there's absolutely nothing wrong with them. I mean, there was one case years ago where it was a little old lady, to be honest, who came in with something like a 2.5 red blood cell count, when average for a woman's about 12, and she came in complaining that she was a little bit tired, and we couldn't even believe she was upright and walking, let alone a little bit tired. But never underestimate the survival and carrying capacities of a little old lady.
Yeah.
Well, I mean, this is excellent stuff. But we're just coming up to our sort of the end of the 45 minutes. So I guess to sum up, I mean, do you know if there's any? So presumably there are research projects out there doing this very thing and coming up or trying to come up with an artificial blood. Do you know how far? Well, we're obviously a hell of a long way away if we haven't got any further than when bulking up with saline.
To be honest, John, I mean, like I say, I've been working in this field basically since I left university, and there's been a headline every couple of years: artificial blood is just around the corner.
It's like fusion.
Just like fusion. The last one, I believe, was the Daily Mail, always believable, from last year, who said it was just around the corner. But then, less said about the Daily Mail, the better, really.
Yeah, and do you know where the sort of the centres of this centres of excellence for this type of thing are? Where what countries or what?
The main, we live in an international world now, but the main have been coming up when I'm doing my research seem to be American companies. Maybe that's just because it's going to be such a lucrative thing in the States, or because they're the only people with it. Normally in the area of biology, we're the world leaders in almost every area, so it's quite unusual not to see us, carrying it through. But it's looking like the Yanks have got the hit on it at the moment.
Were you following the developments in the Japanese one, Riken Institute? There was a thing last year. That was the stems turning stem cells into red blood cells in an acid bath. That had a bit of a tragic end to it, that story, because they were quite a prestigious institute, and obviously the Japanese are, all about their prestige, and it was a bit of a generalization, but it's a culture of honor, isn't it? And basically, it long story short, the study failed to replicate. So, in other words, it was poor science. And one of the authors of the paper that had claimed that they'd done it, he killed himself, and the other lead researchers just recently resigned. I think it was last month. So that was a bit of a disappointment, because that is one way to approach it, isn't it? The stem cell approach.
Yeah, I mean, I'm just reading here, Richard. A group in Madras in India claim they're able to generate a quadrillion. I have no idea what a quadrillion is. It's 400 million or something like that. I don't know. Numbers too for me. Yeah, quantum amount of zeros. But anyway, they reckon they're able to generate a quadrillion pure red blood cells from 1 million stem cells collected from umbilical just a few weeks, and apparently they've patented it, and they're looking to an international patent. Could be something to keep an eye on. They says they're looking at mass production. So there's a lot of clever people doing stuff like that, and India at the moment in particular is really rushing forward in the field of biology. So we'll see what comes of that one.
Excellent. Well, that's I guess a positive note to end on.
John, can I just put a little disclaimer on the end that I'm very much representing myself and my own opinions here, and not the opinions of my employer or the National Health Service in general.
No,
just in case they fire me.
Yes, yeah. Well, no, that was brilliant, Jack. That's a cracking 45 minutes. It went back so quickly. I think we are all considerably more informed about what is a very day-to-day function, blood passing through your body, and I think we're all definitely more informed. But we've hit our time limit now, so we're coming to the end of the episode 17. As always, if you want to get in touch with us, we have various means to do this, and one of them is via our Twitter handles that are on our little panels. One of them is via email, which is thewondrousstuff@gmail.com, and of course you can come to our blog, which is thewondrousstuff@blogspot.com, where you'll find show notes, and Richard is compiling a full topic history of everything that we've covered, and links directly to the YouTube article that they're in, or video that they're in. So until next time, we wish you all a goodbye, and again, thank you very much for watching. See ya.
Bye.
Cheers. See ya.
Bye bye.